This section is going to cover the citric acid cycle. I am going to try to point out maybe a few things that you haven't thought about or learned about in other classes. We're going to start the kind of big picture idea is that this two-carbon group here is actually going to be carried on acetyl-CoA. As an acetyl group, it's bound to a coenzyme, and this is going to enter the citric acid cycle as a two-carbon group, and we're going to go from a 4-carbon group that's within this cycle. We're going to build it up into a 6-carbon group, and then throughout that process we're going to lose a couple of carbon dioxide molecules, so very highly oxidized carbon, and for both of those, and then we're back to a 4-carbon group. We get some energy out of this, we get some reduced coenzymes, and we're going to talk about what we do with those reduced coenzymes. One thing I did want to mention as well is that we have that pyruvate. Remember the pyruvate molecule that I want you to be familiar with? That is going to be converted into acetyl-CoA. But first, we have to get that pyruvate into the membrane inside the mitochondria where all of the citric acid cycle process happens. So we actually have the phosphoenol pyruvate. We can convert that into pyruvate. And that we have a transporter on our membranes in our mitochondria that transports pyruvate through into the mitochondrial membrane. And then pyruvate can be converted into the acetyl-CoA that I just talked about. And that is that 2-carbon source that we're going to use to enter the citric acid cycle. So these, in eukaryotes, pyruvate must be transported into the mitochondria. This is the first step that happens inside the mitochondria. Actually, while the pyruvate's after it's transported inside that mitochondrial organelle, we have the carboxylic acid here on top, and we have a ketone here in the middle and a methyl group on the bottom. So the difference here is that carboxylic acid got replaced with a coenzyme A on the top and a sulfur group. And so this is going to be this is catalyzed by a large complex of enzymes. So the pyruvate dehydrogenase is actually not just pyruvate dehydrogenase, but there's E1, E2, E3 subunits. There's multiple different steps of this. So I'm showing you that here it's made up of multiple complexes. So they have E1, E2, E3 are all made up of all of these different things that allow this process to occur. And for these next 2 slides, I'm going to actually walk you through these different E1, E2, and E3 catalyzed reactions. In this first step, we start with pyruvate, carboxylic acid, and ketone in the middle. And we have a thymine diphosphate, or TDP. And this is derived from B1, vitamin B1. It's a prosthetic group. And a carbanion forms on vitamin B1 ligand. So this is a coenzyme. We have this carbocation down here. And so that's a negatively charged carbon that's not very happy. It doesn't want to stay that way generally very long, but it's going to be very attracted to that partially positive carbon group on this ketone here. So we're going to have this negative charge be attracted to this positive, and as you can see, that results in the bond formation that we see over here. So this is a very temporary bond formation. We see that this group then also, because of that electron associated with that carbon, these electrons actually pop off and leave this form this carbon dioxide group, and then these electrons can jump up into that oxygen that are more electronegative, and we can also see that we've added a hydrogen here, so we can have an alcohol group forming. So now we have this hydroxy ethyl group that is attached to that vitamin B complex coenzyme. And now we bring in this E2 subunit. This is going to catalyze this next step, acetyl-CoA dihydrolipoamide S-acetyltransferase. So the lipoamide is right here, and we're going to be breaking this disulfide bond. So what can happen in this step is this carbon can release the electrons to the oxygen here, and that will then allow it to then release this hydrogen, which can be attracted by the sulfur, and that this bond will then break, and we'll have this one of these bonded to a hydrogen, and then we'll have electrons that are flowing there. They'll be then attracted to this carbon and bond in there. So you'll see that that is what actually is forming here. This double bond is formed once these electrons move towards there, and we can actually have this kind of this thiol group with the sulfur and the carbonyl group attached. So acetyl dihydrolipoamide group here that we have this, and then this is reforming. That B vitamin is reforming the coenzyme, and then now we have this acetyl dihydrolipoamide. So this is the secondary catalyzed step. And we're starting here again, and now we have finally the acetyl-CoA. This is going to be transferred. This thiol group is going to transfer that acetyl group off of the thiol, and it's going to transfer it to the coenzyme A here with the acetyl group. So we're going to be transferring a substitution, essentially a reaction, where we then transfer that group from here over to the acetyl-CoA group. And then we have reduced these sulfur bonds completely. Those can then be recycled. We can also then, so E3 converts E2 back to the original state. So this is the E2 here, and then E3, as I mentioned, remember these are reduced, then they can be oxidized back here into this reformed bond, and this uses FADH. So you can see this FAD becomes reduced into the reduced form FADH. So this is a reduction oxidation reaction. And the E3 subunit of this enzyme complex that's bound to the reduced FADH2 coenzyme then can go through a redox reaction with NAD plus and form one NADH, which is then, so one for every conversion of this whole process, we actually get one NADH out that's formed. So here's the overall picture of this reaction. So we have kind of the big picture. This is this big giant subunit that we have E1 subunit, E2 subunit, E3 subunit, all shown here. Pyruvate, we start with, we lose carbon dioxide, and we form this intermediate, this HETDP intermediate, and this is catalyzed through the E1 subunit enzyme. We then attach that acetyl group onto the sulfur, so the sulfur is broken, disulfide bond's broken, so that we can form an acetyl group. That is then interacted with the acetyl-CoA. We can kind of then take this HS-CoA and attach the acetyl group to it, so we do that transfer of the acetyl group, and then we have to recycle the E2 subunit, so that goes around and that uses the E3 enzyme, so we can recycle this back to the original form, so we kind of go back and forth and create more of these acetyl-S-CoA subunits. Then the FADH that's attached to the E3 has been reduced. Now we can recycle that as well, and we can get a reduced coenzyme out the end of this. So this whole process, we actually gain a reduced coenzyme out of this. So in the next section, we're going to talk about fats, and this acetyl-S-CoA is really the starting point for fatty acid synthesis as well. So we'll come back to this acetyl-CoA idea. So just know that the important part of this is that it's this 2-carbon grouping with a carbonyl group on the back end of it. So we always kind of see these pictures of the pyruvate getting turned into acetyl-S-CoA, and it's just like a little line there, but I just wanted to show you that that's a little more complicated than just simply one kind of enzymatic reaction. So the important part, again, as I mentioned, we have this 2-carbon group that is going to get attached to a 4-carbon group, and then that's going to give us citrate, which is the 6-carbon group. So this is kind of where the name comes from, the citric acid cycle. We make citrate, and then we kind of go through this process to get mostly the important part that we're getting out are these NADH reduced coenzymes. We do get one GTP molecule per turn, but really we get a lot of reduced coenzymes that we're going to see are going to be important for really generating a lot more ATP than just this one GTP molecule. The net result of the citric acid cycle or the tricarboxylic acid cycle is the generation of 4 reduced coenzymes, 3 NAD and 1 FADH2, 2 CO2 molecules, and 1 GTP. This is also regulated allosterically with ADP. ADP regulates this third step, and it's going to be an activator. So if we have low energy levels, we'll have ADP in our system, and that's going to activate this step. NADH acts as an inhibitor for the same step, and so this is a way that we can regulate. We have these feedback mechanisms and feed-forward mechanisms for regulating at this step 3. And we're going to talk about this as kind of the overall, the 8 steps of the citric acid cycle. Next few slides are going to go into more detail about these enzymatic processes. Here we can see we have a transferase reaction. We're going to transfer this carboxylic acid with this acetyl group. We're going to swap off an acetyl group. We're going to add that. We have the HS-CoA, and then we're going to have the acetyl-CoA coming off and transferring onto citrate. So we're going to go from 1, 2, 3, 4 to a 6-membered ring. So this is a synthetase, actually. So we're going to add these together, and this is the entry of these, of going from 4 to 6 carbon group. We're going to attach that group to that carbonyl carbon here, and that will allow us to kind of bend this carboxylic acid down, and this will turn into the OH group here. And we're going to then also oxidize this carbon further to get that carboxylic acid in versus just what it is, kind of just a carbonyl group now. So there's a lot of processes actually happening in here in the citrate synthase reaction. It's enzymatically catalyzing a number of different things, but we're essentially gluing on this oxaloacetate with this acetyl group here. In the previous chapters, we talked about glycolysis and how citrate was a regulator of the glycolysis. And so if we have high amounts of citrate, that means that we have generated a lot of pyruvate and acetyl-CoA, and acetyl-CoA can then add on with the oxaloacetate to generate a lot of citrate. So if we have high levels of citrate, that means that we have high levels of acetyl-CoA. So if I were going to ask you if it's going to activate or inhibit glycolysis, what would you say? This is high levels, and I will not tell you the answer, but you should kind of be able to work through that and think about that question. Once we have our citrate, then we do a rearrangement where essentially we're going to move around those carboxylic acid groupings, and we move this hydroxide down to this other carbon. So this is just an isomerase reaction, and here we have a oxidoreductase that we're going to use. One of the reasons I know this right away is that we have NADH forming, so this is being reduced, and then that means that this is being oxidized. We can see that this carbon is being oxidized, so we're getting more carbon to oxygen bonds down here. So that's one of the types of reactions. Again, like many things, there are multiple reactions going on each step with these enzymes. So we not only have an oxidoreductase, but we have a decarboxylase or a lyase. That's another type of enzymatic reaction. So two types of chemical processes, enzymatic reactions happening simultaneously with the enzyme isocitrate dehydrogenase. These components of glycolysis, some of them will come up here and block the phosphorylation, which will keep it active longer, and some of these components will come up here and activate the dephosphorylation of this, so it becomes in the active form. So these things are both doing the same thing, they're doing it kind of in a different way, right? It's through the dephosphorylation or the phosphatase activity or the kinase activity of that enzyme. So they're all kind of increasing the amounts, hopefully, of the alpha ketoglutarate that'll be synthesized. So we have another oxidative decarboxylation. So it's an oxidoreductase and a lyase, because the lyase part is because we're losing carbon dioxide. Essentially, this is remember we talked about lyase as being involved in double bond formation. Well, carbon dioxide has double bond forming. And then we also have the oxidation reduction reaction and the lyase. So we have a piece that's kind of falling off, double bonds form, and we also have the reduction. So this goes from alpha ketoglutarate to succinyl-CoA. You'll notice that there is a coenzyme on this as well. This is part of that grouping. So this gets not only does this lose the carboxylic acid, but it gets attached to a coenzyme in this complex. The succinyl-CoA, now we have a transferase or a synthetase, and it's coupled with a ligase kinase. So there are or a transferase. So there's a lot of another multi-step reaction where we still have these kind of groups are remaining the same, but now we're going to, this is kind of the end that's going to change, right? We're going to have a oxygen over here that's going to be formed. We got to get rid of that, right? So we have to add that. We're going to transfer that onto that sulfur group. So we're going to transfer in. We're also going to be synthesizing energy here. So GTP generally in humans or mammals, ATP more in plants here. And so we're gonna synthesize. We're gonna make some energy out of this. We're also going to couple this with a kinase. Once we have this succinate, we have an oxidoreductase. So we have the Q to F, QH2 or FADH2 that's forming. And we have a double bond forming, but it's actually not a lyase. This QH2 is part of the electron transport complex. So the succinate dehydrogenase complex this is membrane bound. So we'll discuss this a little more later. But this is actually a oxidoreductase, not a lyase, even though we have this double bond forming, because we again have this FADH2. So anytime we're making reduced coenzymes, as we kind of know that there is something happening here, we don't really lose any oxygens, or we're not adding anything to this, so we're just considered an oxidoreductase. Here we have now a lyase where we're actually adding water to this double bond. So we have this hydroxide and a hydrogen added onto this side, so the H2O, we have one of the, we just kind of split this up. We have two hydrogens here, and this is the water that's going to be added to the double bond here. So we have a hydrase, which is a lyase reaction, that's going to help us form L-malate. And then we get back up to the top of this cycle through malate dehydrogenase. Again, oxidoreductase, we kind of know this because we're generating reduced coenzyme and we're creating a more oxidized oxaloacetate. Malate's actually this component here gives us a sour taste in unripe apples. It's just kind of an interesting fact here. Fun fact: malic acid was first isolated from apple juice and named after the word, the Latin word for apple, which is malum. So that's where that word comes from. That was kind of interesting. Here's a little picture of an apple just to highlight that fact. So we always have these interesting things going on with the mitochondrial membrane and getting in and out of there to undergo glycolysis and gluconeogenesis. These things happen in the cytosol. Citric acid cycle happens in the mitochondria. So we have to get these things back and forth, and it really happens through the malate actually has a transporter, that allows it to come back and forth. There is not one for the oxaloacetate, so we can't get through the membrane with oxaloacetate. So it actually happens with malate. There's also an aspartate transporter. So these are the ways that we can get in and out of the mitochondria. We can't do that through oxaloacetate. So it has to go around. So before we actually We can make oxaloacetate in the mitochondria, but any of this that comes out, and that's going to be used for gluconeogenesis, for example, actually transported out, and this is another step for regulation potentially. But we also, instead of doing this in the mitochondria, this can happen as this is transported out, and it can be synthesized in the cytosol so that we can go on from oxaloacetate to phosphoenol pyruvate to pyruvate and generate some of the components for new glucose development. Interestingly, there's a lot of similarities between some of these enzymes that work in these pathways. For example, if we looked at that lactate synthesis in our bodies under anaerobic conditions and pyruvate, there's lactate dehydrogenase is the enzyme that regulates this reduction of NADH and the oxidation to pyruvate. And this is kind of what this compound looks like. This is the active site of that, and here is the lactate compound being shown here. That's the substrate. We have the coenzyme form of it over here. And if we actually look at a similar enzyme, malate dehydrogenase, they're really closely related. We have the reduction of NADH. We have the substrate in here. Here's the substrate. It's a little bit bigger, but you'll notice this similar carboxylic acid end here. The difference is we have a carboxylic acid end up here. And so this arginine, we still have arginine 171. So it's even the same amino acid number that's going to be interacting here. It's been changed from a glutamate to an arginine so that it can better interact with this top end here. So there's a lot of similarities between these enzymes. They might have even been duplicated and then had a point mutation, but then been used for something else. So this is how we can actually, our body uses different enzymes and compounds the complexity of what we're able to do by just small changes in amino acid sequence and proteins that then allow it to recognize and bind to some other metabolite and do something with that. So this same process happens, and we're able to now generate oxaloacetate because we had this one little change in amino acid side chain on a protein. So it's kind of the evolutionary procedures for how these things can be created. There's clearly an advantage to that. So you can see again, this is just a different highlighted, zoomed in picture of what I was kind of doing over here. There is a size difference clearly, right, with the lactate versus the malate. And this position 102, the amino acid position 102, these must have been mutated to allow for this larger malate molecule to fit in here, because it clearly wouldn't fit in with that amide functional group sticking down into the region. It wouldn't have allowed a carboxylic acid group to be up in that same space. So although they're very similar enzymes, that steric hindrance of this extra group on top here wouldn't allow this enzyme to act on this other reactant. We've looked at a couple of different pathways and looking at regulation, and I wanted to come back to this kind of the pyruvate dehydrogenase, this compound we see right at the beginning of the citric acid cycle with these two groups. And I want you to think about if these reactants here, NAD or HS-CoA versus NADH and acetyl-CoA, not HS-CoA, how are they going to regulate this? Is it going to be inhibiting or activating the process? So which box is the activator, which box is the inhibitor? Is it 1 or 2? I want you to predict that, and you should have the answer for that before we move forward. Just as we looked at a few slides ago with that other component of the citric acid cycle, that third step being regulated through phosphorylation, this also happens with this complex. Phosphorylation actually regulates if this is in the inactive form versus the active form. And so we can see that these different components that are part of this process can regulate the phosphorylation through the pyruvate dehydrogenase kinase, or the phosphatase. So it's mediated through these phosphorylation events. And we call this a cycle, the citric acid cycle, but it's not always really cyclic. There are several pathways that intersect, that pull things out, that pull things in, right? Actually, the malate, we know that can cross the membrane and then be generated into oxaloacetate. Oxaloacetate can be used to do gluconeogenesis. We can make aspartate, which we also talked about in crossing the membrane in the mitochondria, used for amino acids, urea, different nucleotides, the bases, the nitrogenous bases for different nucleotides. Pyruvate here can kind of feed into oxaloacetate. Fats can feed into the citric acid cycle through acetyl-CoA. Citrate can fatty acids, steroids generated from that. Alpha-ketoglutarate, different amino acids can be developed. Different fat can come in, succinyl-CoA. So a lot of different pathways intersect and come in and out of this as well. So just wanted to point that out, that there's more than just this cyclic nature. There's things coming and going that can regulate the speed of these reactions.